Hi, and welcome to the first University of Washington Center for Cannabis Research public webinar. I'm excited to be the first speaker, and today I'll be telling you how we can optimize the medical properties of cannabis. These are my disclaimers. I'm a professor in the School of Medicine, and I'm also the founder of Stella Therapeutics and have a consulting company. And I will actually be telling you a tiny bit about the work that we did at Stella Therapeutics. And all of the results that I'll be sharing today were published in peer-reviewed journals. By way of introduction, so I've been in the cannabinoid field for 25 years. And as a pharmacologist, I think about small molecule, in this case, delta-9 THC or CBD that are produced by the cannabis plant. And I think about how these molecules travel in our body, depending on the route of administration. And this is the concept of pharmacokinetics that I'll be covering. I also think about how the molecules actually interact with their receptor, their targets. So this is the concept of the key lock and pharmacodynamics that I'll be covering also. And all of these bioactivities of THC and CBD, I will share some results when they are tested in cells in culture, in preclinical rodent models, as well as in humans. So in several different laboratory model systems. The goal of my lab is truly to discover new cannabinoid-based transformative therapeutics for the treatment of devastating diseases. And the two diseases that we are currently focusing on are the anti-epileptic properties of cannabinoids, as well as the properties to directly kill brain cancers. And truly our goal is to bring this research all from the lab all the way to human clinical trials. I am also the founder and director of the Center for Cannabis Research, and I'll be telling you a little bit more about this center and who are the individuals that are involved. And just by way of reference, often at the end of my slides, I have punchlines at the bottom. And this, I have 26 slides. Slide number 1. I think I can do this in about 50 minutes. So the Center for Cannabis Research really has a main goal, which is to foster innovative research and also enable and organize this innovative research. So we were implemented in 2017, and our first goal was truly to outline the mission of the center and identify who are the UW researchers that would be interested in being part of this effort. Our first milestone is that we implemented our board of directors that I'll present in a second. We already had a first retreat in 2018 with some guests from WSU. We also established some partnerships with some key organisms in organizations in the both at UW, the ADAI, or the Center for Cannabis Research at WSU, CC Pro, or even other centers. For example, we have a collaboration with the Cannabis Center at the University of California, Irvine. And truly, our ongoing work during these years have been to implement several partnerships between WSU and UW. And we've implemented a work group that is focusing on specific questions here, the effect of cannabis use on adolescents and the effect on impairment, its impairing effect, and I'll be covering a little bit about this. And this is the first of a series of public webinars, and I'm excited, and I really recommend to you to listen to Dr. Carrie Cutler from WSU when she'll be talking about chronic and acute effects of high-potency cannabis on cognition on May 23rd. So again, our mission is to foster innovative research and establish partnership to solidify and protect long-term policies. Here's our board of directors. You can see that actually it's quite diverse. Also in its areas of interest, we have the School of Law, ADAI, School of Pharmacy, Public Health, School of Medicine, College Arts and Science, and Social Work. And this is quite an area of interest for the University of Washington over the last 5 years. We have accumulated approximately $14 million in NIH grants from diverse, actually, institutes. The National Institute of Drug Abuse is a really big one, and NIAAA. And the topics that were addressed in these grants are presented here. And you can see that there's a really big emphasis on trying to understand the effect of cannabis on adolescents, its interaction with alcohol, its effect on PTSD, and a growing interest of the activity of CBD. So our agenda today is the goal. I'm hoping that everyone who's listening to this webinar will become experts in cannabis pharmacology. So we will start actually with a basic hypothesis as scientists. And we will start with an easy one, which is cannabinoids have a unique medical properties. I'll be going over some background. I'll tell you about cannabinoid biology, what we understand currently from how it's produced by the plant and its bioactivity into humans. What are the effects into humans? I'll be looking at how we can actually optimize the therapeutic properties of cannabinoids, starting from evidence in preclinical settings as well all the way into humans. And to develop clinical cannabis, we need to understand the concept of therapeutic index, which is how can we achieve therapeutic efficacy while having minimal or any side effects. The three areas that I'll be focusing a little bit more into are the potential risks of cannabinoids during adolescence, as well as give you an update on the progress on the therapeutic optimization of cannabinoids for the treatment of epilepsy and brain cancer. And I'll finish with some conclusion challenges and future directions. So I really hope that this background will help you better understand the pharmacology of cannabinoids. So let's start with the plants. So the bioactivity of these two molecules. What is the difference between THC and CBD, and what is currently accepted by the scientific community? Here you have two drawings of the cannabis plants, and truly there's not much difference if you just look at the morphology of the plants. This is actually a legal differentiation between the cannabis plant and hemp, where hemp is defined by a plant that produces less than 0.3% THC, whereas any plant that produces more than 0.3% THC is referred to as a cannabis plant. There is no legal parameters to define how much CBD are in those two plants, which is something that probably will have to be addressed in the future. Here's the chemical structure of those two molecules. Here's THC and CBD, and you can see right away two things. One, that these chemical structures are quite similar, which makes sense because they're produced by enzymes by this plant, by the same set of enzymes, and just a difference towards the end of their synthesis. And another big difference that you can see is that these hydrophobic rings, meaning that these molecules are very greasy, are actually slightly different, with a key difference here where the ring is actually open. And this is probably one of the main chemical characteristics that differs between these two molecules and leads to different bioactivities in vivo. So the overarching understanding of the bioactivity of THC is that it is psychoactive, psychotropic, and therefore what it does is it enhances sensory awareness. We enjoy listening to music, the colors are more vivid, there's an enhanced sensory awareness when using THC. It has some medical properties. Some of the most promising ones are its properties as analgesic. It also is an appetite stimulant. It does have some side effects, and the most predominant one that we are concerned about is its impact on the developing brain, for example, during adolescence. And sometimes it does have actually side effects. Not everybody wants to have undergo the sensation of being high, and therefore that could be actually considered a side effect for some patients, for example. The dichotomy is strong with CBD. For example, it is considered that it does not produce any high effects, psychotropic effect. However, it is even thought to tame the cannabinoid properties induced by THC. So actually a balance between CBD and THC might actually be a really interesting medical approach. The most famous medical therapeutic approach of CBD is its approval by the FDA of its anti-epileptic properties that I'll be covering, but it also has some interesting, promising properties. For example, it can be an anti-inflammatory. And truly, we don't really understand the side effects of CBD at this stage. And there are some evidence that very high concentration of CBD might result in some side effects in some vulnerable populations. So THC and CBD produce basically radically different biological activities, or what we refer to as bioactivities. Historically, there's a long history actually of the use of cannabis. It started 2500 BC, the earliest proof of its use in cannabis medicine. And then in the 1st century, it was also used as a medicine by the Greeks and the Roman empires. Moving forward in the 19th century, in Europe and US, it was used. Dr. O'Shaughnessy is probably one of the first who reported some manuscripts on the medical properties and performed actually some medical cannabis experiments in humans and was able to report them. In the 19th century, it was used for its euphoric effects by several of the famous French writers, for example, and they were using a Tilden solution, which is actually an alcoholic extraction of the cannabis compounds, the cannabinoids, and was used for their euphoric properties. So the alcoholic extraction actually makes sense because since it's a hydrophobic compound, a greasy compound, you need to be able to extract it with alcohol. Moving forward to the 20th century, there's been an increase in the cannabis use during the alcohol prohibition. And then in the 70s, the US Supreme Court declared it a substance 1 controlled substance, cannabinoids, and by definition, when a molecule is on Schedule 1, it means that there is no recognized schedule medical properties for these molecules, which is a scenario that is clearly changing these days. And then in 2017, the National Academy of Science worked and looked at the background literature that was on the medical properties of cannabinoids. And they found clear evidence for its anti-nausea properties, analgesic properties, and it could help actually some patients with multiple sclerosis. But they also identified some risk of cannabinoid use chronically at high dose, which were associated with some mental health issues and some potential cannabis abuse and psychosocial impairments in some vulnerable populations. So truly at this time, we still need to define the medical properties of these compounds. What are their euphoric properties, and to what extent are they intoxicating and impairing? So the THC bioactivity is really broad. One needs to think about this as a continuum. For example, we know that some of the bioactivity of THC can be used as analgesic. It can actually, in some cases, help, at least in acute settings, with sleep impairment, insomnia, help with some anxiety and some stress. Those are some of the potential beneficial effects of THC. It also has some clear harm reduction effects, especially in the context of opioid addiction, where a lot of patients that are using opioids for pain management are able to actually replace opioids by cannabinoids, which have a clearly much better therapeutic index and safety profile than opioids. There's increased evidence that they have some potential harm effects, specifically during embryonic development, and as I will show you, during adolescent brain development. And this could lead to some severe psychiatric disorder, for example, cannabis use disorder in some vulnerable individuals. With regard to CBD bioactivity, we know that it has some strong effect in anti-epileptic, anti-seizure effects. I think one of the most promising future area of investigation will be CBD in autism, maybe even CBD in autism and sleep. There's a very encouraging data that are coming out. It could be used again in the context of harm reduction, replacing opioids by CBD for certain types of chronic pain, maybe more geared towards inflammatory chronic pain. And again, I want to emphasize that we don't really fully understand the side effects that are linked to CBD use, especially when chronically and at high doses, and maybe in combination with other therapeutics. So this is actually a growing area of interest in our field. What we're going to do today is we're going to try to develop a new cannabis-based pharmacological therapeutic approach. So it's a drug discovery project. And truly, historically, it has already been done several times. And here I just cover the example of aspirin, where we isolated the compounds from the willow bark. And then there was a first generation of optimization. And now we have aspirin. But actually, what we are able to do today with our molecular understanding of the target that is engaged by this aspirin molecule, the COX-1 and COX-2 enzymes that are inhibited, we can now actually optimize this molecule and have future generation of therapeutics, for example, that target one enzyme versus the other, and therefore reduce the side effects. Same thing for opioids, where we extracted opioids from the poppy seeds and developed, for example, morphine, that is a very efficient analgesic, but we all know has tremendous side effect, addictive properties. And today, because we understand the molecular mechanism by which opioids interact with their target, we're able to further optimize this therapeutic approach and improve the therapeutic benefit and try to reduce the side effect, and therefore enlarging the therapeutic index. So that's what we're going to try to do with cannabinoids. This will be done in what we called evidence-based drug development, and we're going to try to develop a transformative therapeutic approach. The word transformative is important because the way to think about it is a transformative therapeutic approach is when there is no therapeutic approach that is available for this population of patients, and we are able to actually introduce something that is efficient. For example, CBD for the treatment of refractory seizures. That was a transformative new therapeutic approach. So we're going to combine pharmacology and drug discovery to actually create this transformative therapeutic approach. The first thing we need to do is have a reliable source of the bioactive compound, so maybe extracted from the plant in a very reliable fashion, or figure out the chemical synthesis of those compounds and just synthesize them in the laboratory. Then we need to think about what is going to be the route of administration. Are we going to give it as a pill, as a solution, or as an inhaler? Because depending on the type of route of administration, then we will actually have very different pharmacokinetic profiles. Either a rapid onset that reaches high concentration and then is transient, for example, when taken through the lungs by inhalation, or a much slower pharmacokinetic profile, for example, taken with some pills orally. And depending on the therapeutic effect, if you wanted rapid, short-lasting, or long-lasting and slow, then we can simply actually use different routes of administration. We then need to go to the molecular level and understand the pharmacodynamics of how this molecule interacts with this target, which is the concept of the key lock. The key is delta-9 THC here, and the lock is the cannabinoid receptor. And therefore, as the cannabinoid unlocks this receptor, then you change neuronal activity, for example. And does this change neuronal activity in neurons in culture as well as in mice all the way to humans? Because our goal is to make sure that we have a good therapeutic index for our therapeutic approach, which is as you increase the concentration of your compound, how will the patient respond? Initially, you'll have the therapeutic effect. And then here you'll have found a dose that actually will help 50% of the patients or help 50% of the disease. And then as you increase the concentration, now you have a very effective, efficient therapeutic dose. And this is a good example because at this dose, we actually have no toxicity. If we further increase the dose of the concentration, now we start to have some toxic effects. So this is the therapeutic index, the difference between this concentration and this, and the wider it is, the safer the compound is. And truly that's what we're doing when we're doing human clinical trials. We're always monitoring how much benefit we can do versus how much side effects are produced in increasing amount of population so that we can actually have a good understanding of its therapeutic properties. This is the evidence-based approach, and this has to be contrasted with what we call practice-based approach, which is a good example with CBD currently, who is legally sold on the market. And instead of actually designing an experiment to see if it works, what we're doing is we're surveying the population that is using this compound. So this is a practice-based approach to see if it has any therapeutic properties. Let's talk about our protagonist. So the cannabis plant, or hemp, as I said, legally. We're going to be going over the cannabinoids and some of the terpenes. So here is the American Herbal Pharmacopoeia, which is a series of volumes that describe each plant that has medical properties. And finally, about 10 years ago, the volume on the cannabis plant was published. So this book actually has all the basics information on the plant, how it grows, its seeds, its reproductive cycle. Here, what I'm showing is a picture of some of the strains that are available in the legal market. And just wanted to emphasize that it has some really interesting names. For example, Foggy Mountain. I imagine that this is a strain that will probably have some sleepiness properties to it. But one thing to emphasize is that because the cannabis has been illegal for so long, all these strains actually were cultivated independently without sharing any results since it was illegal. And therefore, maybe the Foggy Mountain that is present here in the state of Washington is different than the one that is sold in California, just because there was actually no procedure or protocol to grow this plant. So the botanists, the plant biologists are really excited to actually try to better understand the different varieties of compounds, of plants that one can grow from cannabis. And here are the compounds. So here are the phytocannabinoids. So phyto, produced by the plant, but there's also many cannabinoid compounds that are that we can synthesize in the laboratory and are not present in the plant, and therefore I'll be referring to these compounds as artificial cannabinoids. And this is an example. This is compound JWH018, which is a cannabinoid, very potent cannabinoid that is sold illegally, for example, in spice. It's a fascinating plant because the it has a really interesting, very large DNA, meaning that it'll probably have a very diverse types of phenotypes that can be associated with this plant, either very tall or very short. And then the other thing to consider is that this plant produces other bioactive compounds. So the chemotype of this plant is actually quite large. And here are some of the terpenes that are produced by this plant. And you can recognize some of them, the limoncello, for example, that has a lemon kind of taste, and some other terpenoids or beta-carpholinins, which actually has some really interesting anti-inflammatory properties. So truly, you can see how there's still a lot of work to be done to better understand the cannabis plant and what kind of compounds that it actually produces. In terms of pharmacokinetics, so we need to assess what are the different types of products that are out there. So for example, we can consider the fact that there are some, the original joints back in the days had between 5 to 10% THC. Today, actually, it's closer to 20 to 35% THC in those. So very sharp increase in the amount of THC in those joints. There's some new devices that have been developed. The vape pens contain 70 to 80% of THC, and even the dabbing of resin can reach really high concentration of THC. Or if you go orally, there are some capsules, typically 10 milligram per dose, some edibles that have in some candy form or some drinks, or even in the illegal market, which is actually really concerning. Some of those products can reach actually 100 to 250 milligrams in one setting. And if they're used in one shot, can result in very severe cannabinomimetic effects in humans. So depending on the route of administration, either it goes through the lungs. So if it goes through the lung, you'll see that it actually first goes to the brain directly. So there's no metabolism. Whereas if it goes orally, if it's taken orally, then it'll go through the digestive system. And very importantly, it'll go through the liver, which has the first step of metabolism. And therefore, it will actually metabolize part of the THC. And then eventually, it will go to the brain. So depending on the route of administration, you can have very different pharmacokinetic profile. Here's an example of an individual who is taking THC through the lungs at different concentration, maybe with different actually types of device. And initially, you can see the amount of THC that is in the body. So this is time, and this is the amount of THC in the body. You can see at low concentration, this individual will reach the high sensory awareness and some euphoria. If you use much higher concentration of THC, so much higher amount, you reach a different type of bioactivity, which may actually impair the individual, slow reaction times, or even poor judgment, which is actually something that we have to consider. Or even more higher concentration of THC might reach some toxic effects. And some individual describe very high concentration of THC as a new term called to green out, which is similar to or reminiscent of blacking out with alcohol, where you've had so much drug that there's a period of time where you actually black out. So there are instances where individuals that take high concentration, high amounts of THC, they can actually green out. And this is a new phenomenon that we need to pay attention to. By a different route of administration, now we're actually going to go through the oral route. You can see similar types of pharmacokinetic profiles, just a little bit more delayed, and probably a need of higher concentration of THC because we actually have the first liver pass. And all of these effects, again, are not similar between individuals, and therefore there's quite a diversity in how individuals respond. And I believe that there's a population that is vulnerable to some of the toxicity effects of THC that we have to actually identify. In terms of pharmacodynamics, how does the molecule interact with its, how does the key interact with its lock to change neuronal activity? So it is known that THC activates two types of receptor, mainly the cannabinoid CB1 receptor and the cannabinoid CB2 receptor, whereas CBD activates a different type of receptor, which is called GPR55. So already here, you have a dichotomy in their bioactivity when thinking about the pharmacodynamics, the key lock effects. Those cannabinoid CB1 receptors are expressed mainly at high concentration in the brain, and you can see that they're expressed in the cortex. This is some molecular imaging of the cannabinoid receptor in healthy human individuals, and you can see that there's quite a bit of CB1 receptors in the cortex, which makes sense with the sensory awareness. Some of the cannabinoid receptors more into the basal ganglia, which makes sense with the motor behavior impairment properties of cannabinoids. But cannabinoid receptors are not only in the brain, they're also throughout the body. And actually, there's a big dichotomy between the CB1 receptor and the CB2 receptor, where the CB2 receptors are actually mainly expressed by immune cells, whereas CB1 receptors are expressed by neurons. So there's actually a really great therapeutic opportunities to create some anti-inflammatory drugs that target the CB2 receptor and do not activate the CB1 receptor and therefore do not produce psychotropic effects. These receptors in our body are normally engaged by endogenous ligands, by endogenous cannabinoids. So here's the molecule 2-AG. This is the second endogenous ligand that has been discovered. And you can see here that it's a fatty acid. So this is also a hydrophobic molecule, which makes sense because it binds to the same site on the cannabinoid receptor as THC. So what THC is doing is basically hijacking this endogenous system, the same concept as with opioids, where you have morphine, that is the exogenous drug that acts on the mu opioid receptor, and our body produces endorphins, and what morphine is doing is hijacking this system, producing analgesia. So probably one of the role of the endorphins in our body is to control pain. Maybe one of the role of the endogenous cannabinoids in our body is to also control pain, maybe through different mechanisms. So all of these molecular mechanisms are really important to better understand what are the multiple receptors that are engaged by these molecules if we want to optimize the small molecule for therapeutic properties. What does THC produce in humans? What are the behavioral effects? And what we refer to is that this is truly a polymodal response in humans. And this is the description of what those behavioral effects are in individuals that don't chronically use THC and actually have some prior experience. So there's some somatic effects that you can actually see here: palpitations of the heart, increase in heart rate, a floating feeling. There's some perceptual effects. So here's where the euphoria is. One of the most famous one is the loss of time sense. How long does 10 minutes seems like it's 1 hour. An increase in body awareness, some of the sensory awareness changes, and some cognitive changes. Changes in introspective state, rapid flow, loss of concentration. And this is one of the biggest actually side effects of chronic high dose of THC is disruption of memory impairment. Some individuals actually become anxious under the influence of cannabis. So this is something to also consider. So we really need to better understand the behavioral effects of THC in humans as well as CBD, which has a very different, actually, polymodal response. The main vulnerable population that we're paying attention to are adolescents. And this is because the brain development is actually a vulnerable stage of maturation, where actually all these neurons are trying to actually establish some strong synaptic contacts with the targeted brain areas. And this process starts at the age around 5, is still ongoing during adolescence, and is almost done at the age of 20. And this is because the cells that are involved in creating and developing the brain express cannabinoid receptors, and these CB1 receptors will regulate cell proliferations, how much cells will be produced. They will also regulate how the neurons actually are migrating towards their targeted brain area, and how the neurons are actually establishing strong synaptic contacts. And all of this mechanism is actually guided by the endogenous cannabinoids that guide these neurons. So imagine that an adolescent brain is regularly flooded with THC at high concentration. THC will bind to the cannabinoid receptor and prevent the endogenous to actually guide these neurons, and therefore there can be some dramatic rewiring that happens in this adolescent brain. And one of the major consequences that is currently has been established is that some individual develop some psychotic episodes or even become schizophrenic. So we have to actually start thinking about what is the effect of these compounds on adult cognitive function. And this impairment of brain development is actually more pronounced when you have high potency THC products that have a rapid onset. And this actually has been identified as a new disease in the last decade, which is now called cannabis use disorder syndrome, that is developed by some of these adolescents that are more vulnerable and have this cannabis use disorder symptoms. And here you can look, you can read some of those symptoms: craving for the drug, difficulty to control or cutting down, need more and more potent. So this is really along the lines of an addiction, an addicted type of medicine. So this is one area that my laboratory is really focusing on, because we believe that we need to better understand what this effect is of THC on the vulnerable population, maybe to identify actually some therapeutic approaches, or at least some preventive approaches to try to reduce this effect. So the way we do this in the lab, this is some work that we're doing with my close collaborator, Ben Land. What we do is we, for example, develop some edibles for rodents. So we take some gelatin, and so these gelatin shots actually contain some THC, and we can measure over 1 day how much of the gelatin these mice are eating. So they love to pick at this little gelatin, so they're eating it during the day. Here's the control. And you can add some CBD, they actually also eat. You can add some morphine, and you can add some THC in this gelatin. And you can see actually that the mice, or the rodents in general, do eat the THC gelatin, maybe a little bit less than the normal gelatin, which is something that we're looking into. So the way we've done this is that we've taken some adolescent rodents, either mice or rats. We expose them to THC gelatin during adolescence for 3 weeks, 3 times a week for 5 weeks. Then we stop their consumption for several weeks and look at what is their behavior in adulthood. And what we found is some clear changes in brain anatomy of those adult rodents that consume THC during adolescence, including big changes in the ventral tegmental area, which is one area that produces dopamine, that's really involved in reward type of associated behaviors. And when you test these adult rats that have consumed THC during adolescence in a reward paradigm, for example, their ability to press a little lever for a sucrose reward, and we kind of look at which actually, what is their behavior. What we found is actually the adult rodents that have used THC during adolescence have a much more risky associated behavior, showing that there's been probably a impairment, a change in the reward system, in the reward network that involves dopamine in adulthood. So chronic use of high potency 2-HC during adolescence has an impact on adult reward behaviors. We have some really exciting new tools that allows us to actually better study the effect of THC on the developing brain. This is a collaboration that my laboratory has with the laboratory of Mike Brukus and Larry Zweifel here at the University of Washington. And this is, we have actually a tool that is a fluorescent cannabinoid CB1 receptor, meaning that here is the CB1 receptor, that this is the lock that will be engaged by the key, some of those agonists. And at the bottom of this receptor, there's a fluorescent protein that has been associated. So here are some experiments in cells and culture. So here's the receptor at the edge of the cell. And if you add the agonist, then you activate the receptor and you increase fluorescence. So we can see activation of CB1 receptor in real time, just as an increase of fluorescent light, either in cells and culture, and we can do pharmacology. They actually respond in a dose-dependent manner, so that makes sense. But we can also do it in vivo. Here are some of the receptors that are expressed in the mouse brain, and some of the results that have been published show that if a mouse is on a running wheel and we actually measure the fluorescent signal in its mouse brain, we can see that there's a change in the activation of the CB1 receptor when the mouse starts to run, and a reduction when it stops, showing that the CB1 receptor in a living mouse that is running has been activated by endogenous cannabinoids. So we can actually use this tool to better understand how THC and the endogenous cannabinoid modulate CB1 receptor in the mouse brain. So those are really exciting novel tools that we're currently working with to visualize CB1 activity. So in terms of therapeutic approach, so it's been known for a long time that actually cannabinoids can reduce seizures. And it's been known both from basic science evidence all the way to its FDA approval. And so the initial evidence started with actually THC, where THC has been shown to reduce seizures in rodents in the 70s. And in the 2000s, then the field had started to explore the anti-seizure properties of CBD in rodents. And we can easily measure seizures by EEG recordings in the mouse, and we can actually measure those and see the effect of our cannabinoids on that. But truly, this therapeutic approach was developed in reverse, meaning that the field first heard about the CBD anti-seizure properties in humans. And this is the example of Charlotte Figi in Colorado, where although there were some preclinical results that clinical reports that showed that cannabinoids have some anti-seizure properties in humans, it's truly the story of Charlotte, who had more than 100 seizures per week, and was put under and was starting to use CBD, and showed that actually CBD treatment reduced those seizures all the way to like 1 or 2 per week, which is a dramatic improvement. And this actually started what is now known by the Charlotte's Web, and stimulated the establishment of evidence-based clinical trials. And even though eventually we also started to validate this in clinical rodent models. For example, in some of the work that I've done in collaboration with Bill Catterall here in the Department of Pharmacology, he developed a mouse model of Dravet syndrome, a devastating epileptic disorder in young adults. And this is a mouse, and so it gets these seizures. And you can see as you increase the dose of CBD, you can actually reduce the seizure duration. So CBD works also in our mouse model of epilepsy. So combine all these results eventually led to the approval of Epidiolex in 2018, a very big landmark for the cannabinoid field to develop a novel transformative therapeutic approach. I believe that this is very exciting, but I think that CBD can probably be more optimized, and we can probably either by optimizing its regimen or by even optimizing its chemical structure at the pharmacodynamic levels and optimize the anti-seizure properties of CBD. So although we've done reverse science, what I call reverse science, which is we listen to what the patient are telling us and then we validate it in laboratory, I think now our understanding is at a stage where we can do forward science, which is use our understanding to actually develop novel therapeutics for humans. One of the most exciting new area of research of CBD is probably in the realm of autism. For example, these patients with Dravet syndrome, as they actually have a large amount of seizures during development, the individuals in later times actually develop severe mental disorder, including autism spectrum disorder. And what we found in our mice, this is one of the behavior that we study in our little Dravet mice. They actually have behavioral impairments that are autistic-like. They do not like to interact with an unknown mice, the social behavior. So here you can see a wild-type mouse that actually interacts a little bit more than average with the novel mice. Here's a wild-type mice. CBD doesn't affect that. Here's the Dravet mice that doesn't interact with this mice particularly more than an empty cage. And you can restore this social exploratory behavior in the Dravet mice by providing acute small amount of CBD, a very exciting potential new therapeutic approach for this devastating disease. Other examples are the effect of CBD on sleep that appears to have really promising effect on several stages of sleep, on anxiety, at least some types of anxiety and in acute, and maybe in the context of cannabis use disorder, since cannabidiol can actually temper the harmful effect of THC. So those are areas that we're all exploring. So I think we can still, there's a lot of room to optimize CBD for multiple therapeutic approaches. I've mentioned the endogenous cannabinoids. So what do we know today about the endogenous cannabinoids? So here I'm going to zooming into a part of the brain with two neurons and maybe an immune cell, and a neuron that is connected through another neuron through this synapse, and have a little close-up here. So here's the presynaptic terminal. Here's the postsynaptic terminal that receives the neurotransmitters. And if we think about the first step, which is as you have neurotransmission, for example, activation of glutamate receptors, you activate the postsynaptic neurons, activate these enzymes, the lipases that will release the endogenous cannabinoids, the greasy molecule from the membrane, and then those endocannabinoids will act on CB1 or CB2 receptor, and THC will be hijacking this mechanism at CB1 and CB2 receptor. Activation of CB1 receptor on presynaptic neurons inhibit neurotransmission. So this is a negative feedback loop. Endocannabinoids on CB2 receptor block immune response or anti-inflammatory. So this is another feedback loop. And by contrast, if we look at CBD acts on a different target. So GPR55. So you can see that it will actually modulate neuronal activity through a very different mechanism of action. So these are the endogenous cannabinoids that are acting on their target, and those are the phytocannabinoids that hijack this system. And what we're trying to do is maybe target the endogenous cannabinoids. So maybe develop a novel therapeutic approach by having an inhibitor of, for example, one of the enzymes that eliminates these endocannabinoids or hydrolyzes it. Therefore, this would actually induce the accumulation of the endogenous cannabinoid and maybe have some of the properties, therapeutic properties, that are achieved by hijacking those receptors with THC or CBD. So this is what we've been doing in my laboratory. So cells produce the endogenous cannabinoid. What about this enzyme that inactivates these endocannabinoids? One of the enzymes that we have discovered in the lab is called ABHD6. So this enzyme takes the endogenous cannabinoid and hydrolyzes it. And we have several inhibitors of this enzyme so that we can accumulate endocannabinoids. And what we found is that this approach actually has some anti-seizure properties. So here's a mouse that has some seizures. You see here the seizures is prolonged. And when you treat these mice with an inhibitor of ABHD6, you see that the pattern of the seizures is quite reduced, and even the duration. So this could be an interesting therapeutic approach. And this is the quantification, where you actually have the full susceptibility of the seizures. You treat with this inhibitor, you reduced approximately 50% of the seizures. And actually this works through a completely different mechanism, which involves GABA-A receptors. So we're really excited about trying to better understand this mechanism to further optimize this therapeutic approach. So there's more receptors and more mechanisms that are still there to be discovered and targeted for therapeutic promises. One of the most exciting topics that I've been working on for several years is the ability of THC actually to kill directly tumors. It's been known since the 70s that actually THC can directly kill tumors. However, the effect of THC was not really pronounced, at least in these types of cancer. For example, the lung tumors. It only inhibited 50% of the proliferation. Whereas you don't want to only inhibit the proliferation, you want to kill the tumor, you want to eliminate it. And in mouse model, really, it didn't really work that well, and even at very high concentration. This paper that came out in 2000 is truly a landmark paper showing that actually cannabinoids can kill directly, are much more efficient at killing glioblastomas. Here's a rat model of glioblastomas where we've injected the GBM into the rat. And then this has been treated with THC. So you see a complete, actually, disappearance of the tumor, which is remarkable. Here's a Kaplan-Meier survival curve. So here is all the rats that were implanted with a tumor. And under control conditions, you see all of these rats actually succumb to the tumor. But if you treat them with THC, you can see that there's a delay in the progression of the tumor. Not much, but there's actually some individuals that are the tumor has fully disappeared. And this is actually even better with artificial cannabinoids, for example, the Wnt compound. So this actually posed the hypothesis that we've been working on for about 2 decades, which is, how can we optimize the chemical scaffold of cannabinoids, these artificial, to even further improve this therapeutic approach? And what is the mechanism of action so that then we can truly embark in a drug discovery project? And this is the work that we've done at Stellar Therapeutics between 2011 and 2017. You can actually learn more about Stellar Therapeutics on my laboratory website. And the idea was that, okay, so we have a chemical structure, we have a chemical scaffold. We know that it activates CB1 receptors very well. It's an agonist also at CB2 receptor, and it's not very good at killing tumor cells. So our medicinal chemistry effort has been to modify this molecule so that we can greatly improve its anti-tumor properties and not be associated with any psychotropic effects on CB1 receptor or anti-inflammatory effects on CB2 receptor. We don't want to reduce the inflammation, as actually inflammation might help with the removal of the tumors. So here's the several regeneration of ST Stellar Therapeutics compounds, and our most recent one, ST401, is quite potent at killing patient-derived glioblastoma cells. So here are 4 different types of patient-derived glioblastoma cells in culture. And here's the potency of these molecules, the IC50. And you can see that ST401 actually kills those tumors quite nicely at very high potency in the 50 nanomolar. So what we've done is, this is an artificial small molecule, ST401. It's not been extracted from the plant. We've actually modified using it. We modified it by using medicinal chemistry. We've reached nanomolar potency at killing tumor cells. It passes the blood-brain barrier, which is an enormous advantage for these therapeutics, obviously. It has a promising safety profile in rodents. So our hope is that it will actually also show us a good safety profile in clinical trials in humans. It turns out that this is a novel mechanism by which this compound is killing tumor cells. Thus, the word transformative therapeutic approach comes to mind. And this is one of the big efforts that we have in my lab, is to try to better understand this novel mechanism by which this compound is killing tumor cells. And the main indication that we found is it kills tumor cells, glioblastomas, but other really devastating cancers, including melanomas, thinking about brain metastases, and also colorectal. And we actually recently published some of those results. So we believe that we're on the path to actually develop a novel transformative therapeutic approach and anti-cancer approach. So to summarize, what I've shown you today is that we need to start thinking about evidence-based development of novel therapeutic approach. Instead of doing a reverse reactive listening to what happens to our patients, we now have a solid scientific foundation to actually move forward and do more of a predictive research using evidence-based approach, maybe monitoring what individuals are currently how individuals are responding to the products that are currently available. And this is actually an effort that we're doing with the Center for Cannabis Research with all our partnerships. So there's basically three ways one can think about optimizing. Either you optimize the molecule itself with different types of formulation, different types of routes of administrations, maybe a pill, a little patch on the skin, a slow release device. Another way to think about that is to actually target the endogenous cannabinoid signaling system instead of using an exogenous approach that has very clear advantages. Or the other one is to embark into a medicinal chemistry effort and start with the scaffold and optimize it all the way so that we can actually have completely different types of bioactivities. All of this research has been done in a very defined legal framework. And I just want to cover that real quickly to kind of give perspective of the work that we've done. So again, we have the phytocannabinoids and the artificial cannabinoids. These are controlled under the Schedule 1 license still today. So all cannabinoids that have been derived from the plants are controlled 1 substance, even some of the synthetic compounds that produce psychotropic effects. Over the years, our drug, the supply of our compounds has come from the NIDA drug supply. It has some advantages and some clear limitations. One of the big limitations is that it's the sole source of cannabinoid compounds that we can test in our laboratory. And sometimes actually the quality or the reliability, how often we can actually get those compounds, is not optimal. So I think there's quite a bit of optimization there that can be done to provide researchers with better products that actually reflect more the products that are currently available on the legal market. And actually having a Schedule 1 is quite an arduous process and extensive legal restriction. There's a lot of paperwork that has to be done. So it's really not an efficient mechanism. So truly what we need is we need to relieve these challenges for research so that we can actually move this field forward. And we need to explore new legal pathway that will enable and foster cannabinoid research and development. In other words, today, because our universities are funded by federal grants, it is illegal for us to actually research cannabis based products from other sources. For example, the compounds that are sold and produced in Washington state. We have to go through this process through the NIH. So this is something really that would require to be restructured. And we need to increase the number of studies that we're doing, both in human, in preclinical and basic research, and hopefully increase the amount of funding that is available for this research, since it is becoming an increased question that everybody's actually exposed to and being and questioning. So just to finish, I want to highlight a couple of items. So I think truly optimizing cannabis, the medical properties of cannabis is all about how much dose is given, a small dose versus a big dose. What is and aligned with that? What is the potency of the product? Is it a high potency product, a low partial potency products? What is the regimen? Is this compound, are these compounds going to be provided several times a day or maybe once a week, which will determine the therapeutic index? What kind of device is used? Is it a rapid onset or a slow release device? And who it will be taking? What is is it a healthy individual? Is it a patient that is already weakened by their disease? What age? Is there a particular vulnerable population? In closing, I just want to thank all my past lab members. This is about 20 years of research, my current very talented lab members. And then if you have any questions, please don't hesitate to email us. So you can follow us on Twitter, or you can check out our website that will have actually the recordings of all the webinars that we have done. And in closing, I'd like to thank you so much for listening all the way to the end of this seminar. And you all take care. Take care. Bye.